So here we're going to talk about uncouplers of the electron transport chain. And uncouplers, the meaning is to cause the dissipation of the proton gradient without the generation of ATP. So in essence, an uncoupler of the inner membrane of the mitochondria is when you allow the hydrogen ions to leak back into the matrix without going through ATP synthase. And this in turn will not generate ATP, but rather will potentially generate heat, for example. And so these uncouplers can be natural, and we'll talk about some natural uncouplers, or they can be types of toxins or poisons. Chemicals can cause uncoupling, as well as certain proteins that are made by the body, such as therminogen. And so in this case, what we're going to have is you're going to use oxygen and increase your rate of oxygen usage in order to maintain the electrochemical gradient to make ATP for the cells. So with every metabolic a cell undergoing metabolic reactions, there's going to be some leakage of protons across the inner membrane. And this is natural, occurs all the time. So about 20% of what is called the resting metabolic rate is energy required to maintain the electrochemical gradient due to this leakage of the protons across the gradient. And this is just a global proton leak. Now sometimes, in some cases, the body does want to force the leakage of the protons. And so in the cases of children and infants in particular, they have brown adipose tissue. And brown adipose tissue will have many mitochondria. And the purpose of this mitochondria is to keep the baby warm. And so there is a protein called therminogen, UCP1, uncoupling protein 1. And this is going to cause uncoupling of the electron transport chain in the mitochondria so it can give off heat. So the major function of this brown adipose tissue in infants is to provide a non-shivering thermogenesis because babies are small and they need to maintain their body temperature. And so in essence, what's happening here is therminogen is going to incorporate itself into the inner mitochondria membrane. And as your electron transport chain of the baby is pumping hydrogens into the inner membrane space, therminogen allows for an alternative route for the hydrogens to re-enter the matrix of the mitochondria. So the hydrogen will pass through therminogen instead of going through ATP synthase. Now, norepinephrine is going to be made in response to cold, and this is going to activate a lipase that releases fatty acids. Fatty acids, of course, is going to be able to be oxidized. It's a fuel. And this is also going to activate therminogen. And so, again, therminogen in this case is going to act as an uncoupler and is going to help to transport the protons from that inner membrane space that were pumped by the electron transport chain back to the matrix without generating ATP. But in this case, you're wanting to generate heat. So there are some natural uncouplers. We have uncoupler protein 1, therminogen, which is going to be mostly in brown adipose tissue. We have uncoupler protein 2, which is going to be found in most cells. Protein 3 is in skeletal muscles. We have 4 and 5 in the central nervous system. And all are going to be associated with heat release from fuel oxidation. Protein 2 and 3 also has some protection from oxygen damage, free radical oxygen damage. Now there also are ionophores, and these are referred to as chemical uncouplers. They're going to have the same effect as therminogen, but they are of course not natural. They are something that you are exposed to, and it can cause uncoupling, which typically can have a disease symptoms because it's not something the cell wants to do. The cell doesn't need to make heat, it needs to make ATP, but these chemical uncouplers are going to prevent that. So dinitrophenol is a chemical uncoupler, and this is going to be a lipid-soluble chemical. So that means it can move across the membrane, and it's going to carry hydrogens across the membrane. So basically, remember the hydrogens are creating a gradient, a proton gradient, and you're going to have higher levels of hydrogen on the intermembrane space and lower levels on the matrix. So the uncouplers are going to pick up the hydrogen and diffuse it back across the membrane, bypassing ATP synthase. Now, if you are constantly exposed to dinitrophenyl, for example, and it continues to decouple the inner mitochondria membrane, and you lose that membrane potential to make ATP, the mitochondria integrity and function can be lost, and the mitochondria can cease to function properly. Now, another uncoupler is valinomycin. So valinomycin is going to have potassium. So it has a hydrophobic core with a potassium center. So in this case, it's not necessarily bringing across hydrogen. Instead, it's bringing across another positive ion, potassium. So in this case, you then have your inner membrane space, which has high levels of positive charges, and your matrix has high levels of negative charges, and this creates the proton motive force to generate ATP by bringing the positive hydrogen into the matrix. With valinomycin, you're now going to bring potassium, and so it's going to dissipate those charges. And so your matrix no longer will have that huge negative charge because the potassium will be brought across the membrane, and the positive potassium will dissipate the electrochemical gradient across the membrane. Valinomycin is an antibiotic, and so as we know, microbes also do the electron transport chain, and it's a treatment for that.